I'm Esben from the photonics group of the PhLAM laboratory, and I'm coordinating this project with Luc Buet from GPark. And we are going to pool our competences in optical fibers for my part and neurophysiology for Luc's part in order to develop a miniaturized imaging tool in order to study neurons in deep regions of the brains of small rodents, like the hippocampus, which is hidden under a dense layer of opaque tissue. If you want to study cells in their natural environment, that is to say in the tissue of living animals which could be moving around, then you need to use a microscope. But the tabletop microscope that we know is a bit too cumbersome to study moving animals. So for that we have miniaturized microscopes, which is based on a quite simple idea. It's just to take all the elements of the tabletop microscope and shrink them down as much as possible. If you do that, you can end up with a microscope which is fully functional, but which is only the size of roughly my thumb. This is so small that you can actually fix it onto an animal and let the animal move around and keep on studying the cells in their natural environment. Now, evidently, the smaller the instrument, the better, because the less it will disturb the animal. But it turns out that it's very difficult to just keep on shrinking the optical elements below thumb size. Fortunately, there's another approach, which is to use an optical fiber. The entire optical system of the microscope, we are going to keep it remote from the sample, and we are going to use the optical fiber and bring it into contact with the sample and let light propagate through the fiber to the optical system. Now this achieves the goal of miniaturization because the tip of the fiber is very small. It's almost as small as a human hair. But it comes with the challenge that the light propagation in the fiber is not the same as in free space. So when light comes out of the fiber, we cannot use lenses to recover the image. You have to use some other element, and this element is called a spatial light modulator, which is a pixelized programmable phase mask. And if you know the current state of the fiber, you can program the spatial light modulator with the phase mask that inverts the propagation in the fiber to recover the image. The ESEED is financing a doctorate student who will be working for 3 years on this project. The doctorate student will design and fabricate an optimized optical fiber. And the ESEED is also financing a spatial light modulator, which the student can use to make an implementation of this lensless endoscope.